Hello friends, myself BP Shah, PGT Biology, Kendriya Vidyalaya Kesapuram, Delhi. Today I am continue my tomorrow chapter, that is photosynthesis. In photosynthesis, we have seen that various type of product is formed. In simpler way, we can now we are able to write that during the photosynthesis, the carbon dioxide, in the carbon dioxide, carbon oxygen by 2 hydrogen bond and it combined with water. We can write water in this way. Firstly, tomorrow as we have written, the carbon dioxide combined with water, it form carbohydrate C6H12O6 plus O2. Now, H2O is released. This has been already proved by Rubin and Kamen, Von Nell and Melvin Kelvin. Now, when we balance this equation, this is 6 carbon dioxide, 12 H2O, it gives 1 molecule of glucose, 6 oxygen and 6 H2O. Now, when we divide all these equation by 6, we can find that 6, 2, 2, 1, 1. Now, this entire reaction we can simplify this. CO2, 1 molecule of CO2, it combined with 2 molecule of H2O, it form CH2O O2 plus H2O. So, when we can simplify this carbon dioxide here and 2 H2O, 1 H2, 1 HOH, 1 HOH, the product will form, the product is, we can write with another that is CH2O, that means here, and the another product like oxygen which acts as a byproduct, we can write in this way, and the third one product, that is H2O, this H2O here. So, the entire reaction we can summarize here, that is the carbon dioxide it form CH2O, carbohydrate, oxygen as a byproduct and water. So, this reaction tells us this carbon dioxide, it is going to be reduced. It takes place in absence of light. So, it is called dark reaction. And this oxygen release during the photolysis of water, as I have already discussed. So, this reaction or the product formed in the light reaction. So, with the help of this diagram, one can easily understand how this reactant converted into this product by this way. Now, the question is, what are the product of light phase of the photosynthesis? Yesterday, I have already discussed the during the light phase, that is the phosphorylation, it form ATP, adenosine triphosphate, that means adenine, sugar and 3 phosphate molecules. Adenosine triphosphate and the NADPH2. This we have already told, this is called assimilatory power. So, the answer of this question, what are the product of light phase of the photosynthesis? The ATP and NADPH2 combinedly it is called assimilatory power. It is used in dark reaction. Next question is, what happened to the oxygen produced? We know that oxygen plays vital role in respiration. The plant release oxygen and the animal as well as human being consume this, and this oxygen break down the glucose molecule in aerobic respiration, and the product is carbon dioxide, water, or in case of aerobic respiration it gives 38 ATP. This ATP is used in various metabolic process. So, here the answer of these two questions. Now, as I have already left certain things, that is, there are two effects given by immersion. Immersion first effect, that is called red drop effect, and immersion second effect, that is called immersion enhancement effect. What is this effect? From this diagram we have already discussed the particular pigment that like chlorophyll A, chlorophyll B and carotenoids, this receives particular wavelength of light. When immersion gave certain wavelength of light in increasing order, he found that beyond the red wavelength, when this graph shows, he gave certain wavelength of light in increasing manner. He find that rate of photosynthesis increases, and at the point of red it going to be also increases. When this wavelength beyond 700, greater than 700 nanometer, he found that rate of photosynthesis suddenly dropped. So, this effect is called red drop effect. Immersion gave the first effect, that is called red drop effect. In his second experiment, he added certain low wavelength of light with beyond greater than 700 and wavelength of light, nanometer wavelength of light. He found that rate of photosynthesis again increased. That means, when there is a combination of shorter wavelength as well as longer wavelength, the rate of photosynthesis increased. That effect is called, second effect, that is called immersion enhancement effect. So, two effect, that is the red drop effect and immersion effect, that shows the photosynthesis required lower wavelength as well as higher wavelength. When only we gave the higher wavelength, the rate of photosynthesis decreases, because beyond the 700, that is infrared, plant or the pigment system can't accept this and the rate of photosynthesis decreases. This experiment, how we can perform it? Without the chloroplast, we can't perform, without the chlorophyll, we can't perform this type of effect. So, we have already discussed in my previous classes, according to the Hill reaction, where the chloroplast is isolated, and in chloroplast Hill gave only the light and water, and O2 is released. How chloroplast is isolated? There is a demonstration of the Hill reaction. According to the Hill, he isolates the chloroplast, he take certain leaf like this, any green leaf like spinach, mango leaf, and he detach the leaf from the veins, veinless leaf. He taken veinless leaf, vein is removed, and only the green part which have large number of chloroplast, this, then he homogenized these fresh green leaves with cold 0.25 molar sucrose solution. After homogenization, filter this suspension with the help of filter paper, then again centrifuge it, the filtrate, remove the supernatant, we can easily get the chloroplast, and after getting chloroplast, we demonstrate that the during the Hill reaction oxygen is released. For this demonstration, the Hill used a small amount of oxidized blue color dye, that is DCPIP. The full form of DCPIP, dichlorophenol indole phenol, into the suspension of chloroplast. Chloroplast already filtered, and in this suspension he added DCPIP, and the entire material, entire experimental material, exposed it into the light. This DCPIP, as we have already told, this gives blue color. After exposing in light, this blue color disappear. The blue color disappear. This is because this DCPIP becomes colorless, being reduced due to the photolysis of water, because in Hill experiment he used only water and light and the chloroplast. When this chloroplast added with DCPIP, this water is going to break down and electron, this hydrogen reduce the DCPIP and it becomes colorless. So, by this way we can demonstrate the chloroplast and photolysis of water. In class 11th there is a experiment separation of different type of chloroplast pigment. As we have already discussed in my earlier classes, the chloroplast have different type of pigment like chlorophyll A, chlorophyll B, carotenoids, xanthophyll, student may ask to separate this pigment by paper chromatography. For this same procedure as we have discussed, take the leaf and its vein and veinlet is removed. Take it in mortar and pestle, this type of vessels. This is the pestles. Take this leaf here and add washed sand, fresh sand, and crust it with the help of pestle. We will find that after homogenize it, add 50 to 30 ml acetone in mortar, grind with the help of pestle and filter the pulp. After filtering the pulp, evaporate this filtrate on water bath at temperature 40 to 50 degree centigrade. When we evaporate it, it becomes concentrated. When it becomes concentrated, then we take chromatography paper of about 10 centimeter long and make a pointed end, this like this, and with the help of a scale we marked from the lower end up to 2 centimeter and mark a point with the help of pencil. After the evaporation, take a small drop of filtrate here at the point we have fixed it. Then this entire thing is dipped in a jar, this type of jar, which contain mixture of petroleum ether and acetone solution with a fixed ratio, that is 9:1 at temperature 40 to 60 degree centigrade. Then add this mixture of petroleum ether and acetone in a fixed ratio. Then we take the chromatography paper and dipped inside the jar in this manner, so that in this mixture, in this solution, our filtrate material is not dipped. Hang it and close the jar with the help of its cover. After some time, we will find that the mixture of petroleum ether, that is called solvent, it is growing up. After 1 hour, we remove the chromatography paper and dry it, sun dry or fan dry. We will find that various types of pigments are there in the spotted manner, different types of spots available at a particular distance. In the uppermost layer, we will find that after the dry the strip, it shows the distinct pigment. The uppermost orange yellow band, this orange yellow band, this is carotene. Next to the orange yellow band, you will find yellowish band. This yellowish band is xanthophyll, and the third band just below the yellowish band or the xanthophyll, there is dark green band. This dark green band of universal pigment, that is chlorophyll A, and the last fourth one band yellowish green band, that is for chlorophyll B. Now, in this way we can isolate all type of chloroplast pigment, with the help of paper chromatography. When you have worked with the chlorophyll A, cut this chlorophyll A and make a solution with the help of distilled water, and you may work with the chlorophyll A. With the help of different types of wavelength, we can find the rate of photosynthesis at particular wavelength. In this way, we will do experiment with chlorophyll B, xanthophyll and carotene. So, in this way different types of chlorophyll pigment can be isolated with the help of paper chromatography. So, student can easily done this experiment, which is in. Now, one major things we have discussed in photosynthesis the phenomenon of fluorescence and phosphorescence. When now we are able to make chloroplast or the chlorophyll, when take a test tube and add some water plus chlorophyll as we have isolate from the leaves. When we see this solution in transmitted light, here is the eyes, we will find the color is green. So, here we can discuss when the chloroplast or the chlorophyll visualize under the transmitted light, when light is going to be transmitted it looks like green, but when this light going to be reflected, here some opaque mediums are there, then it return back and our eyes is here. When this pigment, this solution visualize under the transmitted light, its color look like red. How this happen? So, this shows the phenomenon of fluorescence. What do you mean by fluorescence? When the light is transmitted through the chlorophyll solution, it appears green. If this solution seen in the reflected light, it appears red. So, the re-emission of red light by the chlorophyll in the reflected light, this phenomenon is called fluorescence. How this going to be happened? As we have discussed earlier, when the reaction center or photochrome center, it receive light, the electron is going to be emitted. This electron is emitted this electron when received by the acceptor, then it is going to be another pathway like cyclic and non-cyclic phosphorylation. When there is no acceptor, it is going to be returned back within the 10 to the power minus 9 second. When it return back, this appears red and the phenomenon is called fluorescence. Now, when the energy level is so high, this one is called first excited state. Or its time duration is 10 to the power minus 9 second. When its time is more, it again going to another excited state, that is called third excited state. And in third excited state, it changes their spin. Normally in the spin quantum number, one is clockwise, another is anti-clockwise, but when the time is 10 to the power minus 3 second, it changes their spin, both the spin in the same direction, and when it return back, when there is no acceptor, it also gives the red light, red emission. This phenomenon is called phosphorescence. In both this phenomenon, fluorescence and phosphorescence, the chlorophyll pigments give their color, that is red, but there is the timing difference and electrons affinity in which type of excited state they have reached. When it is in first excited state, the time period is 10 to the power minus 9 second, when another or the higher energy level, that is 10 to the power minus 3 second, these also emit the red light. This is the phenomenon of phosphorescence. So, now we are able to clear our idea about phenomenon of fluorescence and phosphorescence. Now, come to the reaction. After this, these all things are related with the light reaction. After the light reaction, the carbon dioxide now able to convert it into carbohydrate by the process that is called reduction. So, reduction of carbon dioxide into carbohydrate, this is independent to light, there is no use of light. So, this type of reaction is called dark reaction. Or it is also called biosynthetic phase. In this phase, for the reduction we require hydrogen and energy. This hydrogen and energy, which is already made by the plant during the light reaction in the form of ATP and NADPH plus H plus NADPH2. So, this utilized in the dark in absence of light, and this carbon dioxide is fixed into the carbohydrate. It takes place in stroma, no doubt, we have already discussed. Where all the enzymes for the dark reaction are present. This reaction, that is the dark reaction or biosynthetic phase, it has a sequence of reaction. Sequence of reaction was demonstrated by firstly Kelvin in particular algae, that is chlorella, and another algae Cynodesmus. By Kelvin. Later on, the another scientist Benson and Basaham also worked in the same field with the help of radioactive carbon, that is carbon 14. So, this biosynthetic phase or the dark reaction, also called Kelvin cycle or Kelvin Benson cycle, Kelvin and Benson cycle. So, we can say that Kelvin cycle, dark reaction, Kelvin and Benson cycle or biosynthetic cycle. The entire Kelvin cycle or the dark reaction has been divided into 3 main phases, that is whole reaction, as we have already discussed, it is a sequence of reaction, that is first is carboxylation. In this carboxylation, carbon dioxide, that is the raw material of photosynthesis, through the stomata, it received to the mesophyll, it is converted into carbohydrate. It is firstly accepted by one of the 5 carbon compound, that is RUBP, ribulose 1/5 bisphosphate, ribulose 1/5 bisphosphate. This RUBP combined with carbon dioxide with the help of enzyme Rubisco. The full form is ribulose 1/5 bisphosphate carboxylase. The RUBP stand for ribulose 1/5 bisphosphate and CO stand for carboxylase. This enzyme is known as one of the most abundant enzyme on the earth. Because it react with the carbon dioxide and the first reaction is going to be start. So, the first process in the dark reaction, that is carboxylation. The second one reaction, mainly here reduction takes place, that is called glycolytic reversal. We will discuss the glycolytic pathway in respiration, where the glucose molecule is converted into pyruvic acid. Here the name is glycolytic, the same cycle of the glycolysis, that is also called EMP pathway. Here the direction of the glycolytic pathway is reverse in order. So, this is called glycolytic reversal. And third one, that is the major one, that is regeneration of RUBP. This RUBP again regenerate for the pickup of another carbon dioxide, and so the cycle is going on continuously. So, these 3 major steps in dark reaction or the biosynthetic pathway. The entire reaction, it require 18 ATP and 12 molecules of NADPH2 for the formation of 1 molecule of glucose. So, entire reaction or this ATP and NADPH2 it came from the light reaction. So, the dark reaction has been divided into two main group. Yeah, we can say there is two main pathway. One is C3 pathway, another is C4 pathway. What does mean by C3 and C4? As the term indicate, here carbon 3, the number of carbon atom is 3, here the number of carbon atom is 4. In dark reaction, in measure of the dicot, measure of the plant, the first stable product, that is when carbon dioxide combined with the RUBP, the first stable product is carbon 3, that is also called PGA, phosphoglyceric acid. Here firstly the carbon contain 1 carbon and RUBP we have already discussed, it contain 5 carbon. The 5 plus 1 carbon it combined to form 6 carbon. This 6 carbon is keto acid, it is unstable. Immediately this unstable compound break down into the 2 molecules of 3 carbon, that is C3 compound phosphoglyceric acid. So, this cycle is called, when this reaction occurs in a particular plant, first stable product is PGA, that is the carbon 3 compound, that is called, that pathway is called C3 pathway. This pathway traced mainly by the Kelvin. So, this cycle also called Kelvin cycle. So, now we are able to say the dark reaction is called C3 pathway, Kelvin cycle, or the dark reaction, or the biosynthetic pathway. In another cases, it was found that until 60, it is already known that the only the C3 pathway is operated in atmosphere and the first stable compound is C3, but in after the 60s it was found that there is many plants like sugarcane and many monocotyledons plant where the first stable compound is C4 compound. It was first of all given by Hatch and Slack. Hatch and Slack. So, this pathway is called C4 cycle or Hatch and Slack cycle. So, now we are able to understand that the dark reaction have two phase, when the first stable product form, that is the carbon 3 compound PGA, that is called C3 pathway, and when the carbon 4 compound is formed, that pathway is called C4 pathway, or after the discoverer it is called Hatch and Slack pathway. But there is no taxonomic bar, no taxonomic importance of this pathway, as here we have two plant of Euphorbia genus. First is Euphorbia corolata. In Euphorbia corolata, it shows, it gives the first stable product like PGA, that is called C3 plant. In another species of the Euphorbia, that is Euphorbia maculata, here the first stable product is carbon 4 compound. So, this shows C4 plant. So, there is no taxonomic importance that particular genus have C3 or particular genus have C4 cycle. In another cases, the Allotropis semialata, it shows both C3 and C4 plant. It depend upon the ecological variant, that is called, also called ecotype. So, in these cases, when the environmental condition is variant, ecotype is variant, it shows the product like C3 or product like C4. So, there is no taxonomic importance. Now, when we move the carbon fixation, the entire dark reaction we can summarize here. First of all, carbon is going to be fixed. There is the input of carbon dioxide here, it is received by Rubisco enzyme, that is ribulose 1/5 bisphosphate, and 6 carbon unstable compound is formed. This 6 carbon unstable compound break down, it forms 3 carbon compound, that is called 3 phosphoglycerate or 3 phosphoglyceric acid. Okay, this is the first reaction. Now, this 3 phosphoglyceric acid again back, that is the glycolytic reversal, that is also called second phase is called reduction. Here the sugar is formed. After the formation of sugar, there is the regeneration of CO2 acceptor, that is RUPP. So, the entire cycle in this slide we will discuss that is the 3 phases. The first phase carbon fixation, second phase glycolytic reversal, that is also called reduction pathway, because here the reduction takes place with the help of various type of enzyme, and the third one is generation of RUPP, which is the most abundant enzyme. Now, the entire dark reaction here we summarized, that is first of all the RUBP, this RUBP is 5 carbon compound, it is a type of carbohydrate in the ketonic form. This RUBP, as the term indicate, 2 phosphates are there, at 1 carbon is 1 phosphate, at 5th carbon is another phosphate. So, this RUBP combined with CO2, this form unstable compound, that is 6 carbon compound, this 6 carbon compound named as 2 carboxy 3 ketorabitol 1/5 bisphosphate. This is very unstable compound. So, it is breakdown, it is going to be breakdown and for 2 molecules of 3 PGA, PGA phosphoglyceric acid, here the acid functional group is acid and 1 phosphate group are there. So, these are the Rubisco enzyme that help in the carboxylation or the first reaction, most abundant enzyme on the earth. Now, this PGA, now there are 2 molecules of 3 PGA, it combined with ATP, adenosine triphosphate, which is already formed in the light reaction with the help of enzyme kinase. it converted into 2 molecules of 1,3-di-PGA, means diphosphoglyceric acid. The molecular formula is same like phosphoglyceric acid, COOH, second carbon CHOH and third one CH2OP, but here the 1 and 3rd have acid group with P, that is the phosphate group. This 3 PGA converted into 1,3-di-PGA, and here is the use of NADPH2. When NADPH2 is going to be used, that means the reaction will be hydrogenation reaction and the enzyme is dehydrogenase, and this 1,3-di-PGA now converted into, with the help of this hydrogen, it converted into aldehyde. This is the aldehyde group. This aldehyde group have 2 molecule of 3 phosphoglyceric aldehyde. This phosphoglyceric aldehyde with the help of isomerase enzyme, it going to be changed into another molecule that we have discussed in biomolecules already, that is called dihydroxyacetone phosphate. This dihydroxyacetone phosphate with the help of isomerase, it is interconvertible. This 3 phosphoglyceraldehyde and the dihydroxyacetone phosphate, it combine with the help of enzyme aldolase and it converted into fructose 1,6 diphosphate, as the fructose is hexose and it has ketone group, and at 1 carbon and the 6 carbon there is phosphate group. Fructose 1,6 diphosphate is formed, it combined with water and phosphate group is removed, it converted into fructose 6 phosphate. This fructose 6 phosphate here the same as fructose 1,6 diphosphate, but here is certain change, at 1 carbon phosphate group is removed, this phosphate group is removed and ATP is formed, and here is the alcoholic group is there. So, this fructose 6 phosphate, from this fructose 6 phosphate sugar is formed, sugar as well as another molecule fructose is formed, which is the main thing of the photosynthesis. The product of photosynthesis, that is the sugar or starch is formed. After that, this fructose 6 phosphate undergoes regeneration, that it forms the various types of compound, erythrose, pseudohaptulose, 7 phosphate. It again combined with the phosphoglyceraldehyde, ribulose 5th phosphate, then xylose 5th phosphate. This ribulose 5th phosphate again return back to the ribulose bisphosphate. So, the entire reaction we can summarize that the carboxylation phase from RUBP CO2 to the 3 PGA, again the regeneration, again the reduction, that is called sugar molecules, again the regeneration that of RUBP. So, this entire reaction is called dark reaction, and after the dark reaction the carbon dioxide is fixed and carbohydrate is formed, again RUBP is formed. So, again cycle is going on. So, this is all about the dark reaction. Here the many questions arises, what are the differences between the cyclic phosphorylation and non-cyclic phosphorylation, how the ATP is formed, what is the differences between the pigment system 1 and pigment system 2, and large number of questions are there. Now, so we are able, dear student, we are able to answer all these questions related with the photosynthesis. Thank you.